Hello students, welcome to the DG classes of Christchurch Convent School, Gadarwara. I am Poornima Shukla, and in the last video we were discuss about the double helical structure of DNA that was given by Watson and Crick. Now listen, how this DNA is packed in our, you know about that the DNA is present in chromosome present in nucleus, and nucleus is present in the cell. So how this DNA is packed in a chromosome? Listen, DNA is what? DNA is a long chain polymer of over 1 million deoxyribonucleotide units. Okay, example, E. coli means Escherichia coli bacteria. In this bacteria, the length of DNA is 1.36 millimeter, and it contains 4.6 into 10 to the power 6 base pairs. Human genome, it contains 3.3 billion base pairs, and total length of DNA is in the human cell is approximately 2.2 meter. So this 2.2 meter length DNA, long DNA, how it is packed in a chromosome, which are very small in size. That was not seen by our eyes. We can see them with the help of electron microscope. You know, so the basic unit of DNA packaging is what? How the DNA is packed in this nucleus, in this chromosome. The basic unit is nucleosome. Nucleosome was discovered by Outdate in 1975, and it is a semi-cylindrical structure of DNA which was composed of histone, proteins and DNA. So न्यूक्लियोसोम किससे बना हुआ है? DNA और हिस्टोन प्रोटीन से. Now let's see the structure of nucleosome. Nucleosome, it is the model of the nucleosome, and how it was made up of. So that it is composed of DNA and histone protein. So this is what? This is DNA. Okay, and in this the stone proteins are tightly packed in the DNA. Means the stone proteins are wrapped in a DNA, and these stone proteins are what? Stone proteins are positively charged particles, and they are basic proteins of low molecular weight. Okay, and they are of 5 types. That was H1, H2A, H2B, H3, H4. So these stone proteins form the nucleosome model. They are tightly packed with the DNA. That DNA which wrapped the 4 histone proteins, these are doubled and form the 8 histone proteins. So 8 proteins, they are called as the octamer of core histone protein. So this is the octamer of core histone protein, and they are wrapped with the DNA. So it is called as core DNA. The other one nucleosome is attached with the other nucleosome, and this DNA is called the linker DNA, which link the 2 nucleosomes with each other. Okay, this linker DNA, its length is what? That is about 80 base pair long. So in this way the nucleosome model is formed. In this way the DNA is packed in a chromosome. This is the beaded like structure which we can be seen in the chromatin fiber, and you know that chromatin fiber are present in chromosome. So in this way the DNA packaging takes place. Understand it? What is that? That is formed by the nucleosome. So DNA packaging, its basic unit is nucleosome. Okay, in the nucleosome, that is the semi-cylindrical structure formed by the DNA and the stone proteins. And stone proteins are positively charged, basic in nature, and they have low molecular weight. There are 5 types, in which 4 types are doubled and form the core of the octamer, and the other one is H1, which is at the outer side of this octamer. Okay, and they form the nucleosome model. So this is about the DNA packaging. 6 nucleosome again attach with coil together and forms the structure called as solenoid. Okay, so 6 type of this nucleosome, they are attached with each other and form the solenoid, and in this way they are arranged in a chromatin fiber, and this chromatin fiber is in a chromosome. So in this way DNA is packed in a chromosome. Thank you.